The ATP PCR system is an anaerobic energy system that does not require food and is a very quick and fast paced. It only lasts about 2 seconds, and you start with ATP, and then it breaks into ADP and a phosphate through ATPase, the enzyme ATPase, and then the phosphate is going to go back into the cytoplasm, and then the PCR, phosphocreatine, is broken through creatine kinase into a phosphate and creatine, and then the creatine is going to disappear and it produces 1 ATP. And then glycolysis is also an anaerobic process that uses carbohydrates, and it lasts about 2 minutes and is also found in the sarcoplasm. And glycolysis starts with glucose, a 6-carbon phosphate, and it's broken down through hexokinase into a glucose 6-phosphate, and then it goes through an isomerase and it's broken into a fructose 6-phosphate, and then the fructose 6-phosphate is broken down through phosphofructose kinase, and that turns into a fructose 1,6-phosphate. And then the fructose 1,6-phosphate goes through something called aldolase, and it splits into 2, a DHAP and a G3P. And then the DHAP is going to go through something called the triphosphate isomerase, and then that's broken into 2 pyruvates. And so the net of glycolysis is 2 ATP and 2 pyruvate. And then the carbon dioxide from the pyruvate is going to turn into 2 acetyl-CoA and go into the Krebs cycle. And the Krebs cycle is going to make 1 full turn, and it's going to produce, I think it's 3 NADH and then FADH and then 2 ATP. And then the hydrogens from the FADH and the NADH are going to cross the inner membrane space of the mitochondria, and they are going to go into the electron transport chain. And the Krebs cycle and the electron transport chain are both aerobic processes, and they are going to take longer time and it's going to be more endurance space. And then the hydrogen are going to push an electron up to push out 4 hydrogens in the first inner membrane space of the electron transport chain, and then the electron is going to transfer over to the second space and push up 4 hydrogens, and then in the third space the electron's only going to push up 2 hydrogens, and it's going to go through ATP synthase. And also the FADH is going to enter in the second space of the electron transport chain and push up 4 hydrogen and then 2 hydrogen in the third part.